10 students. Today we will do a rapid revision of chapter number 2, sexual reproduction in flowering plant. So we will do a revision of this chapter in very fast manner. Okay. So you know that it is a sexual reproduction. That means here the two parents of opposite sex are involved. It occurs with the formation of gametes. Here the offsprings which are produced, they are not identical but they are looking similar to each other, and it occurs usually in higher order organism. The sexual reproduction is somewhat slow, complex and more elaborate process. So here we have to discuss the sexual reproduction in flowering plants. You can also call it as angiosperm. So how this process is going to occur and which is an organ that is involved in a sexual reproduction in flowering plants. In a flowering plant, the sexual reproductive organ is a flower. We are using a flower in a number of cases. Like for an example, we are using to express our emotions. It's going to have a cultural value, it has a religious value, it has emotional value, and lots of values it has. But for a plant, it is a reproductive organ. This flower that consists of 4 types of a valve, okay, and which are the outermost is calyx. The second one is, it's called as corolla. The third one is called as androecium. The fourth one is called as gynoecium. Okay, so these are the 4 valves. The individual unit of calyx is sepal. Individual unit of corolla is petal. Individual unit of androecium is stamen. Individual unit of gynoecium is carpel. Clear? From this first two, sepal and petal, or the calyx and corolla, they are accessory valves or units. And from this, they are called as necessary, or you can also call it as reproductive valves or unit. Clear? These are the valve and this is a unit. Okay. From this, the androecium is the male reproductive one and the gynoecium is female reproductive one. Okay. So that is about a part of a flower, that which part that involves in a sexual reproduction. Usually these are the two necessary, androecium and the gynoecium, they are necessary part because they involves in a sexual reproduction in a plant. Okay. So these are the part of a flower. Okay. Let's discuss the individual part. First we started with the androecium. Androecium is a male reproductive one and its individual unit is called as a stamen. So let's start with the stamen and later on we will discuss the gynoecium and the carpel. So it's androecium. It's a male reproductive valve. Its individual unit is called as stamen. Okay. Now what are the parts of a stamen? Stamen mainly consists of 2 parts. First, a stalk-like portion. This is a stalk, a long, slender stalk-like portion. This is called as filament. Second, this portion, which is usually bilobe and present at the terminal end, it's called as anther. And this anther and filament, they are connected by connective. So usually stamen consists of 2 parts, anther, that is bilobe, present terminal end. Second is filament, a long, slender stalk-like structure, they are connected by connective. Okay. Now we have to see the internal structure. So we will start with the internal structure of an anther, because this filament is just a stalk-like portion and it is not a fertile part, it is a sterile part. The fertile part means the part which is involved in a sexual reproduction is anther. So let's discuss the internal structure of an anther. When you see the internal structure of anther, so if you take the transverse section, anther will look like this in the transverse section. See, this is our parts of an anther. What it looks like. Clear? The longitudinal groove that is running throughout it. Okay, this is how it's look like. Okay, if you see the anther is bilobed structure. Bilobed structure means it contains how many lobes? 2 lobes are present. Okay, in each lobe, so you can see there are 2 lobes. Okay, in each lobe, how many theca are present? 2. So that's why this structure is called as dithecous. What's called as dithecous? Because each lobe contains 2 theca, so that's why this structure is called as dithecous. Now, if you see the corners, then the corners are how many corners? 1, 2, 3, 4. So they have 4 corners. So that's why this structure can also be called as tetragonal structure. In the transverse sections, the internal structure of anther look like a tetragonal because it has a 4 corners. Each corner it possesses a microsporangium. Each corner, okay, that possesses the microsporangium. This microsporangium is usually circular in outline, means it is a round, it's a circular in outline. Okay, so this is a structure of anther. Now, if we see the structure of this microsporangium, the microsporangium consists of 4 layers. How many layers? 4 layers. The first one is, it's called as epidermis. Second one is called as endothecium. Third one is called as middle layer. And the fourth one is called as tapetum. So how many layers? 4 layers. Epidermis, endothecium, middle layer and the tapetum. From this 4 layer, the function of these first three layers, epidermis, endothecium and the middle layer, the function of it, they help in protection and dehiscence of anther. Okay? The innermost layer is a tapetum and the function it provides nourishment to the developing cells or internal cells which are present inside. Okay, so the function of epidermis, endothecium and the middle layer, it provides protection and they help in the dehiscence of anther, whereas the tapetum, that is the innermost layer, they provide a nourishment to our developing cells. Okay, when this anther is young, in the center of this microsporangium, it contains homogeneous cells. These homogeneous cells, they are called as sporogenous tissues. Okay, or you can also call it, it's a group of cells, so that's why what we call it as tissues, or their cells can also be referred as sporogenous cells. The cells, they are present in the center, which are uniform, and they are called as a sporogenous tissue, or which are homogeneous, they are called as a sporogenous tissue or the cell. Okay, from these sporogenous tissues, now there is a formation of a microspore. So now we will discuss how the microspore formation starts. So before I start, just learn the structure of stamen. Stamen consists of filament, anther. Anther looks like this in the transverse section. Anther is bilobed, 2 lobes are present. Each lobe contains how many theca? 2 theca, so that's why it's called as dithecous. How many corners? 4 corners. Okay, each corner has a microsporangium. Microsporogenesis is circular, consists of 4 layers: epidermis, endothecium, middle layer, tapetum. The function of this first three layers is protection and dehiscence of anther, and the innermost is tapetum. They provide a nourishment. When the anther is young, there is a presence of homogeneous tissue in the center of a microsporangium called as sporogenous tissue or cells. From this sporogenous tissue, the microspore formation starts. So let's see how the microspore formation occurs. See, the process of formation of microspore, this is called as Microsporogenesis. So what is a microsporogenesis? Genesis means formation of microspore. This is called as microsporogenesis. Here the pollen mother cell, okay, this pollen mother cell is a diploid in nature, 2N. This pollen mother cell, diploid in nature, it divides by a process called as meiosis. Okay, and it forms haploid structure. These 4 haploid structures are formed. N. Okay, so this structure which is going to be formed, it's called as microspore tetrad. As it is a meiosis process, obviously one microspore or pollen mother cell, one pollen mother cell generates a 4 microspores or microspore tetrad, and the number of chromosome gets halved. Okay, compared to a parent, the number of chromosome gets halved. Initially, this 4 cell remains attached to each other. When it becomes a mature and dehydrate, this all cell becomes separated. 4 cell becomes a separated. Each one, now we can call it as microspore. Okay, each one is called as microspore. Okay, or this you can also call it as pollen grain, and thus a microspore formation occurs. The process of formation of this microspore is called as a microsporogenesis. If you see the structure of this microspore or pollen grain, then what are the structure of pollen grain? Usually the pollen grain is spherical with a diameter of 25 to 50 micrometer. This is the diameter of a pollen grain. Okay, but it may also be of a different shapes. Like it may also be a oblong, it may be also ovoid, or the other shapes are also present, or disc-like. Such kind of a pollen grain are also present. If you see it's a surface, the surface may be a smooth or spiny. The surface of this pollen grain may be smooth or spiny. Okay, this pollen grain, it consists of how many layers? It consists of 2 layers. Okay, one is called as cell wall, and the second one is called as cell membrane. You can also call it as plasma membrane. The cell wall consists of 2 layers. The cell wall of pollen grain is made up of 2 layers, exine and intern. Okay, exine is the outer layer of cell wall, intern is the inner layer of cell wall. Means the cell wall of pollen grain is made up of 2 layers, exine and intern. The exine is made up of organic material called as sporopollenin. This is the organic material that is too much resistant to any kind of acids or any kind of a strong bases, or it cannot even denature with a higher temperature. So it's a very strong one. Next, the another one is an intine, that's inner layer, and that inner layer is made up of cellulose and pectin. And like other, the cell membrane is made up of lipids and protein. Inside this cell membrane, there is a presence of cytoplasm. Okay. Now, so this is the structure of a pollen grain. If I draw a typical structure of pollen grain, this is a pollen grain. Okay. It consists of how many layers? This is the layer of cell wall. But at some position, the exine is absent. The point where the exine is absent, this is called as germ pore, through which the pollen tube will develop. Pollen tube will get develop. Means this is a place where the sporopollenin is absent, because the sporopollenin is so hard it cannot be broken down by any kind of a treatment. Okay, so that is about the structure of a pollen grain. Now if you see the structure of pollen grain, then the pollen grain is also causing a respiratory distress or respiratory syndromes, because the pollen grain of some plant, like for an example Parthenium or the carrot grass, which have been imported along with a dwarf variety of a wheat, that is inhaled along with air, and that may causes a respiratory syndromes like asthma, COPD, etc., or bronchitis might going to be developed with it. So you have to be very cautious while inhalation. In our Western countries, there is always a prediction of a pollen release. Okay, so they can come to know that how to go outside, and then they do a protections. Apart from that, some believe that this pollen grain can also be used as a rich source of a food, because they believe that they are rich with nutrients, and so that's why it can be used by athletes and the horses in a race. Okay, nowadays in the Western countries, the tablets and syrups and the medicines of this pollen grains are also available. Okay, This pollen grain that is dehisces from anther, releases from anther. Once it is released from anther, it remains viable for how much time? Means it can remain for living how much time? It depends on the type of a pollen grain. Some pollen grain can remain viable for few minutes, and some can remain viable for a few months. Okay, and some can even viable for a few years. Like for an example, in case of a wheat and rice, its pollen grain remain viable for 30 minutes after release. Okay, they can be viable after, for 30 minutes after the release. Whereas in case of certain plants of Rosaceae family, Leguminosae family, and Solanaceae family, they can remain viable for months. Okay, so the viability of pollen grain may vary from plant to a plant, and it ranges from few minutes to few days to few years. Okay, depends. If you want to preserve this pollen grain, you can also preserve this pollen grain at low temperature, and here you are providing a low temperature of -196 degrees Celsius with the help of liquid nitrogen. This liquid nitrogen provides low temperature -196 degrees Celsius, and in this temperature a pollen grain is stored. Such technique of preservation of pollen grain is called as cryopreservation. Okay, it's called as cryopreservation. Okay, and that can be stored in a pollen bank. Okay, as we store money in our bank, similarly pollen can be stored in a pollen bank, and seed can be stored in a seed bank. Okay, so that is about a pollen grain. Okay, the male reproductive part and pre-fertilization process, that means gamete formation, have been finished. Now we will discuss a female part, and you know that it is a female reproductive wall in a flower. The female reproductive wall in a flower, it is gynoecium. This is the female reproductive wall. Its individual unit can be called as carpel or pistil. So those flower which possess the carpel or pistil, they can be called as pistillate flower, or you can also call it as female flower. Okay. Now which are the parts of a carpel or a pistil? The carpel or pistil, they are made up of three parts. First is called as stigma. This can serve as a landing platform where the pollen grain can fall. This is a tube-like structure. This tube-like structure can also be called as style. And the lower part, this is called as a bulge portion. This is called as ovary. Okay. The number of carpel in a flower may be one or it may be many. If it is one, then this flower is called as monocarpellary flower, and if it is many, then it is called as polycarpellary or multicarpellary flower. Okay, that is depends on the number of carpels in a flower. Now this ovary, that ovary contains a one cavity. This cavity is called as ovarian cavity, or it can also be referred as locule, L-O-C-U-L. That locule, it contains a placenta. Okay, and the placenta is attached with the ovules. Okay, so this placenta contains a ovule. This is a oval shaped structure, what we call it as ovule. This is a ovule. The number of ovule within an ovary, that may vary. Some plants are such that, or some fruits are such that which has one ovule. Okay, some are such that which has many ovules. Like for an example, mango, it has one ovule. Okay, if you say wheat, that also has a one ovule. Rice, that has a one ovule. But if you see example of watermelon, papaya, they have many ovules within an ovary. Okay, so the number of ovules also going to be vary. Okay, so these are the three parts of a carpel: stigma, style, ovary. Ovary contains cavity called as ovarian cavity. That contains a placenta. Placenta is attached to the ovule. Number of ovule may going to be vary from plant to a plant. It may range from one to a many. Okay. Now we will see the structure of a ovule. How the ovule look like. This ovule is oval in shape. Okay, and that is attached with a stalk-like portion. This is a stalk-like portion to which it is attached. This stalk-like portion is called as funicle, and the portion of ovule to which it is attached, this is called as hilum. Means the region of ovule to which the funicle attached, that is called as hilum. Okay. Now this ovule, it contains 2 layers, and these 2 layers are called as integument, and that is called as covering of ovule. Covering of ovule, that's called as integument. Now inside that there is a presence of nucellus, not nucleus, it's a nucellus, which is filled with a reserve food material, and then there is a presence of embryo sac. Okay, this embryo sac or female gametophyte develops from megaspore mother cell. The ovule can also be called as Megasporangium. That is another name, Megasporangium. This cell develops into a megaspore, and embryo sac and female gametophyte gets developed. Okay. Now these are the parts of an ovule, brief parts of an ovule. Now how the process occurs? Megasporogenesis. Megasporogenesis means a process of formation of megaspore from megaspore mother cell. So here the megaspore mother cell, a 2L, megaspore mother cell. Okay, they divide meiotically, means a process occurs is a meiosis. So obviously you know that how many daughter cells are produced? 4 daughter cells, each containing half number of chromosome, means 2n number of chromosome, and they are called as megaspore. So one megaspore mother cell generates a 4 megaspore. But from this 4 megaspore, 3 will degenerate and only 1 remains, and that is considered as the only that is remaining. That is called as functional megaspore. From this functional megaspore, whole, you can say that female gametophyte or embryo sac formation occurs. So that's why this type of development is called as monosporic development. So what happens to this? I am just drawing this one, enlarging this functional megaspore. This is a functional megaspore. Okay, this functional megaspore, it divides consecutively 3 times by mitosis, and total cells which are going to be produced, this is a 2nd time division, and this is 4th time division. Third time division, sorry. And at the 3rd time division, all the process that occurs by mitosis. How many number of cells are going to be produced? Total 8 number of cells are going to be produced. 1, 2, 3, 4, 5, 6, 7, 8. Okay. These 8 cells, they are arranged in an embryo sac. They are arranged in an embryo sac in such a way that 3 cells at the chalaza, 3 cells at the micropyle, and the 2 cells which are present in the center. So this 8 nuclei are going to be formed. The 3 which are present at the chalaza, what it is called? They are called as antipodal cell. And the 3 which is present at the micropylar end, they are called as 2 synergid cell and 1 egg cell. Clear? So how many cells are present, or how many nuclei are present at the chalaza? 3 antipodal. And how many at the micropyle region? Micropyle region, that is 3. From this 3, 2 synergid cell, 1 egg cell. And in the center the 2 cells are present. They combine together and form polar nuclei. So now at the maturity, the 8 nuclei structure is going to be formed, which become a 7 cell. 7 cell and 8 nuclei. Okay, ultimately 7 cells are going to be formed at the maturation. 8 converts to a 7 because in the center the 2 are joined together. Okay, so this is a development of, you can say, a embryo sac or female gametophyte and their arrangement. What is going to occur? Okay, just look over it. Okay, you remember which are the cells they are present at the micropylar end and which are the cells they are present at the chalice end. Okay, just have a look. So that's all about a male and female gamete formation process. Now once the gamete is formed, now the next process is start, what we call it as pollination. So what is a pollination process? Pollination, that means a transfer of pollen grain from anther to a stigma. This is called as a pollination process. Okay, the pollination process can be of two types. First is called as self-pollination and the second one is called as cross-pollination. In a self-pollination, the pollen grain is transferred within the same flower or it is transferred to another flower of same plant. This is called as self-pollination. And what is cross-pollination? When the pollen grain is transferred to another flower of a different plant or say another flower of another plant, then it is called as a cross-pollination. Okay, this self-pollination can be of two types. First is called as autogamy, second is called as heterogamy. Okay, and the third one, that is called as cross-pollination. That cross-pollination can be referred as xenogamy. Autogamy is usually found into a bisexual flower where the pollen grain is transferred from anther to a stigma of same flower, same plant. Within the flower, the pollen grain is transferred. So it is very easy to observe in case of bisexual flower. Okay, like for example, the flowers of Apiaceae, Lamiaceae and Cactaceae family, that shows autogamy. Heterogamy, that means a transfer of pollen grain from anther to a stigma of another flower. Okay, from one flower to another flower, but the plant remains same. Within the plant, the pollen grain is transferred. This is called as heterogamy. Okay, so if you see, the functionally it's look like a cross-pollination, but genetically it's a self-pollination. Okay, because here the pollen grain is of same plant, but it is transferred to a different flower of a same plant. And what is xenogamy or the cross-pollination? The cross-pollination, that means a transfer of pollen grain to a different flower, and even the plant is also another plant. Then this is called as a xenogamy or it's called as a cross-pollination. Okay, so this is called as pollination, a different types of a transfer of pollen grain. But here the pollination does not occur as such. That requires agents, and these agents are called as pollination agents. The pollination agents can be of two types. First is called as abiotic, means non-living agents which help in a pollination, and second one is biotic agent, that means a living agent which help in a pollination. In an angiosperm, both the gametes are non-motile, so they need a transfer. And to transfer it, they need a certain kind of a help, and that is provided by a pollination agent. Which are the abiotic pollination agents? Usually there are two. One is wind or air, and the second one is water. Okay. The pollination by wind is called as anemophilic. Pollination by water is called as hydrophilic. Okay, and pollination by insects, it's called as entomophilic. and pollination by animals, it's called as zoophily. So here the different types of pollination agents, abiotic and biotic. Those plants which are pollinated by bee, in their case the pollen grain is such that it is very light, non-sticky, can be easily carried out by wind, and the stigma is such that the stigma is feathery, so it can easily capture, and the stigma is waxy. So if the pollen grain falls on it, it do not allow to transfer to another way or carry by wind. This is usually happen in a grass or castor, maize. Pollination occurs with the help of wind. The pollination which occurs with the help of water is called as hydrophilic, and it is only limited up to a certain monocotyledonous genera. It is only found in monocot aquatic plant, means those plants which found in a water, like for example Vallisneria, Hydrilla, Zostera. Zostera can also be called as marine grass, whereas the Vallisneria and Hydrilla, that is found in a fresh water. And you know that how the pollination occurs in that, because it's important. Vallisneria in hydrophily in Vallisneria, or the pollination in Vallisneria, Zostera, the process is important. Even insects like honeybee, ant, butterfly, moth, normal housefly, they also help in a pollination. So they comes under the insect. And the animals, the large animals like lizards, squirrel, or arboreal animals, they also help in a pollination, that is called as a pollination by animals. That means insects and animals, large animals also helps in a pollination. But you have to remember the two important example. The Amorphophallus, that is pollinated by fly. Yucca plant is pollinated by moth. And I also give one more example, Kigelia plant. Kigelia plant, that's pollinated by bat. Even sometimes the bird also helps in a pollination, like hummingbird and the sunbird, they also helps in a pollination. But you have to remember these three examples. Amorphophallus is pollinated by fly, Yucca is pollinated by moth, and Kigelia, that is an animal, a large animal, that is pollinated by a bat. Okay, Kigelia is a plant that is pollinated by a large animal called as bat. So this is called as pollination agents. And that's all about process called as pre-fertilization. Gametes are produced, male and female gamete, and now they are transferred by pollination, and these are the agents which helps in a pollination process. Okay, so that's about a pollination and pollination agents. Now once the agents help in a pollination, then what happens? Then the fertilization process occurs, because by pollination the gametes are going to be transferred, and once they are transferred and they brought together, then now the process occurs, it's called as a fertilization. Okay, fertilization means a fusion. But how the fusion occurs? To understand how fusion occurs, you have to understand pollen pistil interaction. How pollen and pistil interact. So I am just drawing a carpel. You can also call it as pistil. Okay, there is a presence of embryo sac. I am directly drawing the embryo sac inside it. Presence of 3 cells. These are present at the chalice end, what we call it as antipodal cells. 3 cells which are present at the micropyle. From this 3 cell to synergy cell, and plus 1 egg cell, and in the center there is a presence of polar nuclei. These all are haploid antipodal cell. These all 2 synergy cell and 1 egg cell, they all are haploid, and this polar nuclei, it's a diploid because it is made by fusion of 2 nuclei. Okay, N and N. Once the pollen grain falls on it, okay, so the stigma will decide it whether to accept this pollen grain or to reject it. The acceptance of this pollen grain is occur if it is a compatible pollen. If it is incompatible pollen, then it will be rejected. Okay, if it is a compatible, then it is accepted. If it is incompatible, then it gets rejected. If rejected, nothing process will happen. If it is accepted, then the further process, the interaction between pollen and pistil start. What we call it as pollen-pistil interaction. Okay. The communication occurs between the pollen and pistil, and that occurs with the help of chemicals or the hormones. You can also call it as hormones, by which the interaction takes place. But if it is compatible, then it is accepted and the interaction occurs with the help of chemicals, and there is a development of a tube-like structure. This tube is called as pollen tube. Once the tube is formed, so the content which is present in pollen grain, that is released into a pollen tube. So what is the content in pollen grain? The content usually which is released are 2 male gamete and 1 vegetative cell. Because the generative cell is divided into 2 male gamete and then 1 vegetative cell. Now it grows and it grows towards a micropyle, and it enters towards a micropyle region. See, it enters through a micropyle region. The tube grows. Now whatever the content are present, you know the 3 content. This pollen tube has 2 male gamete and 1 vegetative cell. They are released into an embryo sac. From that 2 male gamete, you know that how many male gamete? 2 male gamete. 1 male gamete, it fuses with egg cell. This is an egg cell, and that leads to a formation of zygote. Clear? Second, the another male gamete, from 2 male gamete, one has been fusing with an egg cell. The another male gamete, it fuses with a polar nuclei. And it forms PEN, primary endosperm nucleus, that is 3N, because the polar nuclei is 2N. And this male gamete are always haploid, you know it. Okay, so that leads to. So here the fertilization, fusion occurs 2 times. In the first time, the 1 male gamete fuses with a female gamete, egg cell, form a zygote, 1 fertilization. Second, the another male gamete fuses with a polar nuclei, and that leads to a formation of PEN. As the fertilization occurs 2 times, this type of fertilization occurs in an angiosperm. It's called as double fertilization. Okay, what it is called? Double fertilization. Clear? And at the end of double fertilization, diploid zygote, 2N zygote, and triploid PEN, primary endosperm nucleus, is going to form. Okay, so this is about a brief revision of chapter number 2. Okay, the remaining topics we will discuss in the another session and the next session. So keep learning this chapter number 2. Okay, thank you for watching.